Good morning, scholars and scoundrels. It is evening, actually late in the night where I am, but by the time you guys watch this, it will be morning. It is Tuesday. I am not at school, as you guys noticed. We talked about yesterday. You guys got a bell ringer at the front of the room. Hopefully, you gave that your best effort. I told Mrs. Sexton to take away cell phones if they were becoming an issue. You'll see why that's going to be even more important in a second here. But as you guys know, yesterday we started cell division, which is also called cell reproduction when we make more cells. We most often call this process mitosis, where we start with one cell, we go through some changes and some chromosome and DNA changes, and we end up with two cells. So, I think Mrs. Sexton does a great job with stuff, but sometimes it probably helps to have me go over it. So I will, right now, as I magnify it here, be going over the bell ringer with you. You should all correct it. You'll see, again, this is going to be really important here in a minute, but I'm going to go kind of quick. So Mrs. Sexton, feel free to rewind if you need to. But you guys all got a bell ringer that says cell division. This was our introduction one. There's one or two tough ones, but you guys should have been able to do this. So when we look at DNA that is unwound, we call this chromatin. Chromatin with an N at the end, whereas when it coils to get ready to become those chromosomes, we call it chromatid. Coiled chromatid. When it's unwound, it's chromatin. If a cell has one set of chromosomes, it is called, that word from yesterday is haploid. Haploid. We haven't learned yet about this, but usually your sex cells are the only thing. For males, that's sperm cells. For females, that's egg cells. Usually those are the only haploid cells because we're passing on one half of our genetic information. Speaking of which, number 3 says human cells that have 46 chromosomes. So we know from yesterday we learned that human cells that have 23 pairs are fairly normal. So this would mean they have two sets, and we know two sets has to do with the prefix di. So if human cells have 46 or two sets of chromosomes, we call those diploid cells. Number 4 says the process where cytoplasm divides. Yesterday we talked about how this is the end of the cell cycle. This is after telophase. This is where the cell has to really pull apart. We have that cleavage furrow. That is where we have real cell movement, and cell movement means cytokinesis. So that's where the cytoplasm splits and pulls those things apart. Number 5 says chromosomes. We know yesterday that's those little strands of DNA we talked about. They are genetic material, so A is true. We know that. B says they are in all living organisms. All living things have DNA. That is true. We also know that they're condensed forms of chromatin that get coiled up to form chromatid, which get together to sister chromatids to form chromosomes. So number 5 should be D, all of the above. Number 6, when we coil that DNA, those special proteins are called histones. Those special proteins we wind it up on, like the extension cords I talked about, are histones. That is spelled Histone. Number 7 is a little bit of a toughie. In mitosis, if we have, not a human cell obviously, but if we have a parent cell with 16 chromosomes, each daughter cell will have how many chromosomes? Lots of traps here, guys, but when we go through mitosis, we copy the cell. So if the parent cell has 16 chromosomes, the daughter cell will have 16 chromosomes because we double it to get it to 32, and then when it splits, each one has 16. So mitosis is making two copies of the same thing. So number 7 should be the letter C. Everybody flip to the back. Yes, that includes you, Caleb, Sam in the corner. Get off your phone, Mario. Third block, Logan, put your phone away. Jackie, turn around. Jose, pay attention. On the back side, number 8. This picture should look very familiar. Yesterday we learned about this picture of a chromosome. So the top box is pointing to these two different strands. These are sister chromatids. Sister chromatids. This middle box is pointing to this special protein that's holding those sister chromatids together. We call that middle box a centromere. A centromere because it's in the middle. And then last but not least, we haven't really talked about what this could be down here, but you could call it chromatid because it's coiled up. You could call it DNA because that's true. What we'll also learn is that a special segment of DNA down here, or anywhere for that matter, is called a gene. That's where we get the word genetics, and your genes are what make you who you are, and what we're really talking about there is the DNA. Number 9 is we're using letter codes here. When the spindle apparatus first becomes available, that's when we're starting to talk about those toe chains. That is prophase. We're starting to get the fibers ready to start to pull things apart. That's the very beginning. It's the preparation. Preparation starts with the P. P is prophase. Number 10, when the chromosomes are at opposite poles. When the chromosomes are at opposite poles, this is kind of a tough one, but when the chromosomes are at opposite poles, that means they've gotten pulled apart already. And if they've gotten pulled apart already, that means we're past anaphase. They're already at the very ends. Just like your grandma's telephone antennas, we're at opposite ends of the TV. Telophase is where chromosomes are at the opposite poles. So number 10 should be T, telophase. Number 11 says chromosomes line up at the equator. This is also called the middle. Middle starts with M. When the chromosomes all line up down the middle, that is called metaphase. We talked about that clue a lot yesterday, metaphase. Number 12, the end result of mitosis. There's a lot of traps here, guys, but we've talked about mitosis. Starts with one cell and it makes two. If you go look, the only one that works for this is D, 2 haploid cells. And they're haploid because they stay the same. 13 and 14, I did not talk about 14 yet, but we did talk about 13, uncontrolled division of cells. This will be a video you guys watch in a second, but when we have cells that divide and divide and divide and don't get told no, stop, that is called cancer. That oftentimes results in a tumor, but cancer is uncontrolled cell growth. We have not talked about this one yet, so I don't want to necessarily give it away, but when we have cancer that spreads from one site to the other, you'll hear about, Lance Armstrong had 2 types of cancer. Many people will, my great-grandpa had lung cancer and it spread to his liver, and then he had liver cancer. When it moves around like that, it has a very special name. I don't want to give it away, but I will tell you, a benign tumor is a lump of tumor cells that aren't dangerous. They're not cancerous. So you'll get reading a little bit, but those of you who might have guessed the letter E would be correct. Okay, and we'll talk about that more later. So your bell ringers, hopefully you got those all right. That is the kind of stuff that will be on your quiz. Speaking of quiz, I have one final announcement before you guys kind of jump into the rest of the day here. Just a little update. You guys know my world's kind of been turned upside down the last few days, but I talked to the other biology teachers, and we have to stay consistent as a department. So on Friday, you guys learned about mitosis to start. You kind of cheated your way through that little cycle page, which you may or may not redo. Yesterday, I was at school. I lectured about mitosis, and we got through most of it. You got pink sheets, which should be very, very helpful. Today, which is Tuesday for you guys, you're going to practice mitosis. I got about 4 different things you're going to do, including a Kahoot that is going to help you master what mitosis really is. On Wednesday, then, you are going to take a short mitosis assessment. I do not want to call it a test because it's probably not as stressful as a test, but it is going to be a pretty big quiz over everything you've learned in mitosis, just like your bell ringer was. And you're also going to get a review sheet. Oops. You're also going to get a review sheet that goes over everything we've learned in the quarter. Okay, so on Thursday, then you'll get some time to make corrections to your mitosis quiz, and then you guys will take a final exam. Now, I know we want to freak out right away. The final exam will be about 30 questions. It'll be about questions about what makes something living versus non-living, or what type of things does a eukaryotic cell have, or how are plant cells different than animal cells. It is all very big picture. Most of the questions are taken off the old test, and the main reason I really agreed to this and bought into what the rest of our department was doing was that test can replace your lowest test grade. Your final 30 questions you'll take can replace your lowest test grade. So for some of you guys panicking about your grades and all that stuff, this is a really good opportunity for you to show what you've learned the whole semester and earn that back. All right, so with that being said, I believe you guys are going to watch a short video over cancer. You're all going to very intently practice mitosis with some worksheets in your packet. You'll notice that not all the answers are perfect. Some of the things are kind of in prophase and kind of in metaphase. As long as you're close, I'm not going to count anything off. You guys should use your pink sheets. You can work with one partner, but watch the cancer video. We'll take some notes on cancer in a second here. And why don't you guys actually go ahead and pause this video for a second and get your packets. And if you guys could, I'd love for you to get your packet. I think it cut off a little bit there. So bell ringer should be passed forward. Mrs. Sexton, pass it if you or pause it if you need to. But everyone, snag your packet and really quick flip to page 21. Page 21. Feel free to pause if you need to. We yesterday talked about cytokinesis. Here's that cell plate we learned about in plants. Here's that cleavage furrow we talked about. I'm not really going to give you guys any formal notes here, but what we need to write under control of cell division is not that important. What you could go right is that there's regulation proteins, and those proteins are what say, hey, we skinned our knee, we have enough skin cells, so please stop. A lot of textbooks refer to those proteins as stoplight proteins because it's red or green. Make more or stop. What I wanted you guys to go right was two quick things here. Where it says cancer on page 21, you guys should all go right uncontrolled cell growth. Uncontrolled cell growth. Uncontrolled cell growth. Mrs. Sexton, pause it if we need to, but maybe even put in parentheses mitosis. When mitosis gets out of hand and just goes and goes and goes, that is what we call cancer. And we know that that's one of the main killers in all the world, which is why we're spending time learning about it. Okay? We know that the true end result of mitosis, oh man, I think the baby's crying, is two cells that are identical, and we call those haploid cells because they have two sets, but they're two identical cells. So, sounds like I gotta go take care of a baby. You guys are gonna watch a cancer video and then do a bunch of practice over mitosis. Please use your class time wisely. It will impact how difficult I make the mitosis quiz. Do your best. Take your time. Ask questions. Mrs. Sexton has the key. Rock and roll, block 1 and 3. Make me proud.